From Microbe TV, this is Immune, episode number 89. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. How are you all doing?
Is it really cold up there?
It's really cold. I mean, it warmed up quite a bit. It was -8 this morning. Then by the time I went to leave the house, it was 0. So that's progress.
You're talking about Fahrenheit, right?
I am. So that is, what is it? Really just cold, really cold.
Right now it's -12 Celsius here. It warmed up 2 degrees. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, great to be here. It's a very tropical 17 Fahrenheit here. But yeah, it's still too cold, very snowy.
And the cold is why we don't have Steph here today, because apparently the school's closed and she was the designated parent there. And so she couldn't join us. I guess she could, but we would probably have screaming kids in the background. And we thought that would probably not be the best. So.
Can you imagine like 20 kids in the background while we're recording?
No, that would not go.
That would be.
I think she only has 2. So
but then you have to take care. No, she did have to take care of her own kids, not everybody else's.
I think she just had to go get them and bring them home or whatever. Yeah, because daycare and whatever else closed.
Mm.
Yeah, it's that time of life when it really
kids
happens.
rule your life. Nothing you can do about it. All right, but we have some cool science for you today. But first, I would like to tell you that if you enjoy these conversations about science, good luck finding good ones going forward. But we're going to always do it.
Yeah.
We'd love your support, financial support. We don't do ads. We don't sell things. We just depend on you giving us financial support. So go to microbe.tv/contribute. I have a snippet in the paper today. We're going to start with Brienne, who's bringing viruses into the equation. Apparently.
I am bringing viruses into the equation. But I decided that I wanted to bring viruses into the equation because this paper talks about an immune mechanism that we don't often think about. It's sort of a not very classical immune mechanism. And it's perhaps one we should be thinking a little bit more about, especially because I know that you often enjoy papers, Vincent, where non-neutralizing antibodies are involved. And this is one example of a non-neutralizing antibody. And
in fact, your year review for TWIV was all the non-neutralizing papers we did. So many.
I know. And usually when I think about non-neutralizing antibodies, there are a specific group of other functions I think of. And this paper talks about yet another function involving a protein called TRIM21. That's not one that many people think of. And so I wanted to highlight that. This paper is from Nature Communications. It was published at the end of 2024, and the title is Antibodies Targeting the Crimean Congo Hemorrhagic Fever Virus Nucleoprotein Protect via TRIM21. The first author is Shanna Leventhal, and the senior author is David Hauman. And most of these authors are from the Rocky Mountain National Labs in Hamilton, Montana, although there are a few from the Medical Research Council in Cambridge in the UK, Johns Hopkins, and HDT Bio in Seattle, as well as National Institutes of Allergy and Infectious Disease in Bethesda as well. So these authors have been working on this virus, Crimean Congo Hemorrhagic Fever Virus. It is a negative sense RNA virus that is transmitted by ticks, usually in Europe, Asia, and Africa, that can lead to fever and severe hemorrhagic disease.
So I have a question. This hyaloma tick.
Yes.
Do we have those? Like, do we have ticks that can spread this? Do you know? No, you don't know.
I don't know.
Okay.
My first thought is, where's Dixon?
Asia, Europe, and North Africa. Nope, we don't have it.
I think we only have, I mean, that's where it's been found thus far. Yes, yet. Okay. They certainly mention changes in this tick's distribution due to climate change.
Yep.
Yeah, someday everything's
here.
going to be everywhere, right?
Well, I guess some things will not be certain places they were before, as it won't be.
Maybe, but everything is going to
anymore,
Ithaca.
But yeah.
Cindy, everything's going to Ithaca.
No, everyone's going to come to Ithaca because we're going to be the only place that doesn't have everything. That's what I think. I'm living here.
They're all going to fall in the gorges on the way.
Maybe.
So this is this virus can cause between 5 and 70% case fatality rate, and there are no countermeasures. It's listed as a high priority pathogen by the WHO. And this group previously made a vaccine against this virus, which was a self-replicating alpha virus-based RNA vaccine that expresses the CCHF nucleoprotein. And this vaccine was protective against lethal disease in mice as well as in non-human primates. And when they tried to determine which immune responses were the correlates of protection in these previous studies, they saw that this protection correlated with high titers of non-neutralizing antibodies, and really seemed independent of T cells and B cells. And really, it seemed to be something about these non-neutralizing antibodies. They also talk a fair amount here about the fact that this antigen is nucleoprotein, which is an internal protein inside of the viral particle that's really encapsulating the genome.
I'm surprised they make it only against NP. Maybe they were trying to make a T cell vaccine and then turned out didn't need T cells.
Yeah, they have two earlier papers, one looking at this vaccine in mice, one looking at non-human primates. And in the earlier paper, they use a few different antigens. So they don't just do nucleoprotein, they use other antigens. And I think that they thought it would be one of those other antigens. And were particularly surprised that it was really the NP vaccine that was protective.
What would happen if COVID had turned out that you needed nucleoprotein and not spike?
Then we probably would have been in bad shape.
We would have
had a
stayed home a
vaccine
lot
that was
longer.
targeting spike,
done that
right?
clinical trial with spike and nope, no protection.
Yeah.
We're lucky.
Yeah.
We're lucky, but we did kind of have some ideas from MERS trials and things that a spike antigen might work.
For that class of viruses, yeah.
Yes, exactly. And so this paper is, like I said, a follow-up of some of their previous work to try to figure out, well, what is this immune mechanism that is allowing for protection in their nucleoprotein vaccine that they've used.
And just to be clear, like when we were generating antibodies to spike protein, because it's on the outside of the virus, it was actually blocking binding to the receptor. So it was preventing the virus from infecting new cells, right?
Right, exactly.
So it's not clear how an antibody, which can't actually bind to the viral particle because the antigen's inside, how that might actually work. It's weird, right?
Right, exactly. And they do a little bit of experiment on that in the supplemental data, where they look to determine exactly where the antigen is being found, and they see that it's largely in the infected cell intracellularly, and they're not seeing it, on surface virions, and although they do see it rarely on the surface of infected cells. And they use that in part of their argument about the fact that there isn't neutralization going on. They don't do any neutralizing assays in this paper, but they are in the previous paper, showing that neutralization is
not the mechanism,
not the main thing. So the first thing that they do here is they do a useful experiment to show, is this definitely antibodies? Are we sure that this is antibodies and not some other immune response? And so they take serum from vaccinated mice and passively transfer that serum into other naive mice, and then challenge these mice who've received the antibody passive transfer. And they show that passive transfer of antibodies is completely protective. And so this is something in the serum, something in these antibodies that's leading to protection with this vaccine. And they now start to say, okay, well, which of the, functions of antibodies, is important here. And so they think about things like Fc gamma mediated opsonization or complement deposition or NK mediated functions. Those are based on their data looking at the localization of the antigen. And because they did see it a little bit on the surface, they thought, well, maybe it could be NK cells, maybe it could be ADCC, things like that. And so they repeat their vaccine study in mice that are either deficient in the Fc gamma receptor, deficient in complement C3, or deficient, or that they have in which they've depleted NK cells. And in all three of those situations, the vaccine worked perfectly fine and was still protective. And so it seems as though antibodies are important, but none of these sort of classical antibody functions are at all involved in this protection. And so they move forward into their next experiment, and they know about this other type of antibody effector function, or that is possible, but again, not very frequently talked about, which involves a protein called TRIM21. And TRIM21 is a cytoplasmic antibody binding protein. And so it allows antibodies to act in the cytoplasm of the infected cell, which is in a lot of ways entirely counter to what we think about with antibodies. We usually think about them acting extracellularly, but TRIM21 seems to be a intracellular protein that
many years
antibodies.
ago on TWIV, we did a paper where antibodies to adenovirus involved TRIM21, cytosolic too, not on the cell surface at all. Very interesting.
Exactly. They mention adenovirus here. Cindy, were you going to say something?
Oh, I was just going to say, and this TRIM21 protein is not to get the antibody into the cytoplasm, because as I understand it, we still don't know how that happens. Is that correct?
Right. Yeah,
but we do know that they can get in. And when they do get in, they can bind this TRIM21, and it somehow is involved in this antiviral effect.
Exactly.
In the case of adenovirus, it dissociates the capsid, interestingly. TRIM plus the antibody dissociate the capsid in the cytoplasm.
And it's dissociating the ones that it's making to make new particles, or because it needs to dissociate to the incoming.
Yeah.
Does it just do it too soon? Like that's the virus goal, is to dissociate its capsid.
Yeah,
so
normally
it can
it's docked on a nuclear pore. And if it's just floating around the cytoplasm, then it's not going to get in.
Yeah, then it can either be detected by innate immune sensors or otherwise degraded if it comes apart too soon. And there are some other kinds of intracellular destabilizing proteins that are also important in other situations. So here they just want to know, is TRIM21 involved in the protection that they see in this vaccine? And so they do their vaccine protocol in TRIM21 deficient mice, and they find that in TRIM21 deficient mice, the vaccine no longer protects. And so TRIM21 is somehow involved in the protection that they are seeing. They also repeat their passive transfer experiments and transfer antibodies from vaccinated mice into TRIM21 deficient recipients. And they see that the recipient also has to have TRIM21 in order to be protected from this antibody response. And so this sort of highlights this TRIM21 intracellular function of antibodies as being really important in this vaccine setting, that they were not expecting, and that, is not one that we normally think about. And they show differences in pathology in the mice, and they further kind of clarify that it is not T cells by depleting T cells, showing that T cell depletion does not influence the vaccine efficacy in these mice. And then they set up a modified neutralization experiment because with a traditional neutralization experiment, you are adding neutralizing antibodies to virus preparation and then looking to see whether that virus can enter a cell. And here, what they will instead do is they open up the cells to let antibodies in, and then look to see if they see neutralization when the cells are permeabilized. They do electroporation to permeabilize cells and allow antibodies in, and then say, if you can let antibodies in and you have TRIM21, do you now see neutralization?
And neutralization in this case means just that the virus doesn't infect those cells productively?
It means that you don't get production of more virus. Okay.
So it's really a replication assay? Is it a replication assay or neutralization assay from the virologists?
So they have a funny name for it. Let me find the name. They kind of think about it as a neutralization assay, or they refer to it as a neutralization type of assay. But you're right; it probably is more about replication, and so they call it an electroporated antibody-dependent neutralization assay. But they talk about the fact that shows that it inhibits replication.
But what's interesting, though, is if even for antibodies that we usually consider neutralizing antibodies, tell me if this is correct thinking, that when one runs those assays, they're taking the virus and incubating with the antibody, then adding the whole gamish onto the cells, right? So it's probably the antibody in excess. So it's possible that some of these antibodies could actually be going in and functioning by this mechanism, but you're not seeing it because you're assuming that they're working by blocking receptor binding.
That is correct. I guess in a normal neutralization assay, you cannot exclude this.
Okay.
From happening.
It could be multiple mechanisms for sure.
They do in this paper, do the control of what happens if you don't electroporate.
Okay.
And they don't see anything if they don't electroporate.
Oh, okay.
So you have to have the electroporation in this assay in this experiment, in order to get the blockade.
They are measuring infectivity.
Right.
They do a TCID 50, a 50% dose. So they're looking, it's kind of a traditional neutralization, except they have to electroporate the antibodies into cells, which is weird.
Yes. And it doesn't work if you electroporate the antibodies into TRIM21 deficient cells.
Yep. Need TRIM21.
So you need TRIM21 and you need the antibodies to get into cells some way, which, as Cindy points out, we don't know what that actually is physiologically. I doubt that we are all electroporating or electrocuting ourselves in order to get the antibodies in now.
And please don't go do that.
No, please don't try.
It's not going to help.
But there's always some infectivity. The antibodies in the TRIM don't get rid of all of the infectivity.
They don't.
No.
And I wonder also with that, they're using things like mouse L929 cells for that. And so I don't know that those are the most physiologically relevant cells either in terms of what you would actually see in an infection here. But it looks like this intracellular blockade by TRIM21 is in fact what is responsible for the effect of this vaccine. And so I think that tells us some interesting things about Crimean Congo hemorrhagic fever virus and how we might want to act against, intervene against it. But it also reminds us that there are antibody functions that we still don't really understand. You can hear just from our discussion, we don't even know how the antibodies really get into the cell. They don't explore here how that antibody is blocking this virus. And that, could certainly be subject for a future paper.
So they didn't look at all about like what you guys were saying about adenovirus, where it's a disassembly thing?
They did not look at that.
Okay.
And part of me wonders how much of that is related to challenges of working with this virus versus adenovirus.
I think, is this a BSL-4?
I think it is.
I would think so.
That's
And
why
did
Heinz
they use
Feldman
actual
is
virus?
Yeah.
Yeah, Heinz Feldman, he's up at he runs the BSL-4 at Rocky Mountain Lab, so
Okay.
Right.
probably that's why.
Yeah, they use the virus
They
for
do.
these studies. It's wild.
Very
Because
nasty.
most people will. Well, I mean, in theory, I guess you have to because of its mechanism of action, right? You can't really do. People will do pseudoparticles if they're looking at some sort of neutralizing antibody function.
Right. But since you don't know what the function is, you can't do that.
So you need the whole. They won't have the genome, right?
The pseudotypes will only have the glycoprotein, right?
Well, you
might,
can't put
right.
the nucleoprotein in as well. It won't function properly, right?
No, you can't really. That's very specific.
Yeah.
Because it has to bind to the nucleic acid, right? Which is only going to work for the same virus. So the pseudotypes, typically you put the glycoprotein in, say, VSV, right? But you couldn't put the nucleocapsid into the CCHV nucleocapsid into VSV. It's totally different.
Yeah.
Unfortunately, yeah.
So this is it seems to be an important mechanism when you isolate it. But you said they did do a prior study where they were testing different vaccine candidates, and this was the most effective one?
By far the most effective one.
That's crazy.
So when they looked at the other, they did either nucleoprotein, glycoprotein, or the two combined, and it really seemed like nucleoprotein was the only antigen that was giving them protection.
It's really interesting because imagine the virus particle in the cytoplasm, and then there's these antibodies against nucleocapsid protein, and then TRIM21. Nucleocapsid proteins on the inside of the particle.
Right. So it's probably not about uncoding kinds of things. It maybe TRIM21 is like directing it into a different intracellular location for degradation or something.
Yeah, it could be that. It could be that these virus particles are taken up, say, by endocytosis, and then they fuse, and now the nucleocapsid RNA is in the cytosol, and that's the target of the antibody in TRIM21. Most likely, yeah.
For the assays, did they actually look at production of virus, or did they look at copies of genome? Because
no, they looked at infectious virus.
Because could it be that it is somehow affecting the exit, so it needs to assemble the viral particle, and if the antibodies are there, they're interfering with assembly because if your nucleocapsid protein is wrapped around the DNA, it needs to get stuffed into the capsid. It won't fit with the antibody, right?
Totally reasonable, yeah.
Maybe.
I don't know what their timing was here. It depends when they put the. I think they electroporate the antibody, then they infect. Is that right, Brian?
They do.
So.
I'm looking. So it looks like they electroporate, and then they give 24 hours of recovery to the cells, and then they infect.
And they infect.
Yep.
I wonder if somehow the antibody being recognized by TRIM21 activates some defense mechanisms that work to help limit the virus if it infects, like interferon production or something like that.
They do show, they have a supplemental figure that says that this is not type 1 interferon dependent.
Okay. Yeah.
But yeah, I think that there's still probably a lot that they could look at.
They measure viral replication at 72 hours post-infection. So, Cindy, totally could involve assembly blocks. Yeah. Yep. When you're waiting that long, sure.
But I think that just reminding ourselves of the other mechanisms that aren't front and center in the textbook, and how important they might be, and Vincent's favorite non-neutralizing antibodies.
This is very cool.
Were things that deserved a snippet.
And I'm guessing that, this made it in a good journal, but I'm guessing that people who work in this area with these antibodies that get in the cytoplasm get a lot of flack because people are like, how can there be antibodies in the cytoplasm, right?
Yeah.
And we'll probably eventually identify a protein that moves those antibodies into the cytoplasm, and everybody like, oh, of course, this was an obvious mechanism, right? But it just is one of those things where you have observations you don't have a full explanation for, and a lot of people are just like, oh, that can't be real, and just dismiss it. But yet, if you push and push, you can find out something really cool and different.
And I think the other thing I like about this paper kind of on that front is they really do systematically go through the possibilities and show, okay, yes, it happens with transfer of antibodies. No, if we use all of these different knockouts for things that would be the usual suspects, they don't matter. And so it does sort of go in a nice fashion through some of the possibilities to show that, yes, this TRIM21 mechanism is what's going on.
Yep.
So they call TRIM21 a cytoplasmic FC receptor.
Yep.
So basically that's how the antibody binds to it, right?
Yep.
It's got an FC and it's floating around in the cytoplasm. So I wonder what its function is. Is this it?
Yeah, I mean, in theory, this might be it.
Interesting.
Yeah, I don't know.
I'm guessing it probably makes some sort of lattice or complex or something.
Maybe. I'm thinking a little bit about TRIM5 and HIV, and they're probably not super related, but I know that at least part of that is about directing things to the proteasome in addition to changes in uncoding. So
That's cool. Now we have 2 examples. So maybe this gets into my class now.
One's not enough, but 2. It's
well, you just never know if it's a little detail, right?
Yeah.
You want to be principle driven, and if you give them too much information, they freeze, right? So.
Yeah, I think that this is a paper that sort of my students could sort of go through the logic of and understand pretty well too on it. So
yeah,
cool.
Very good. Like that very much. Okay. I just ate lunch, Cindy. Is that relevant?
It's very relevant. So I picked a paper that is very, very different from what Brianne picked. The title of this paper is called Identification of Antigen-Presenting T-Cell Interactions Driving Immune Response to Food. The first author is Maria Caniso. And the senior authors are 2, I believe. It's mostly Gabriel Victoria, who we've spoken to before, and Daniel Masita. So the point of this is starting off by, Vincent, you just said you ate. So do you consider food foreign or not foreign? So would we consider food part of our self or something completely foreign?
I would say it's foreign.
Okay.
Right? So if it's like plants and whatever else, it's not me. It's not me.
It's transient, right? It goes through, but generally I think I would call it foreign. Brianne?
I specifically tell my students when I show them a picture of eating a burger and tell them that I'm not a cow.
No,
so I tell it foreign.
Definitely not a cow. So, but think about this. If this is something foreign and it comes into our body, why don't we fight it? Like, why don't we react to it? Do we react to it?
Some foods you do react to.
Some foods you do react to. Well, okay. So just because they give you a tummy ache doesn't mean that you're having an extreme immune response to them. But it's a good question, right? So what exactly is happening between us and our food? And the idea here is that there's some sort of tolerance mechanism so that we're not continuously reacting to the things that we eat. And this is, I think, really interesting because there's a lot in the news and the popular media about different kinds of diets and should you eat this, should you not eat that. There's things about fermentable oligosaccharides and short chain fatty acids and gluten. And, there's entire grocery store aisles, at least at our, grocery store locally. It's an entire aisle that's dedicated to gluten-free foods, right? So there's a lot of people thinking, oh, I shouldn't eat gluten. And should you or shouldn't you? I mean, there's definitely people who should not eat gluten, and because there is actually a disease called celiac disease where people develop an immune response against the gluten and it causes massive inflammation in the intestines. So yeah, in that case, you don't eat gluten. But what is gluten intolerance? Like, is that a real thing? Should people eat? Should they not? Is there gluten sensitivity? It's all kind of murky. For what I know. But basically the bottom line is that some people will develop true allergies, like peanut allergies, for example. And those are type 1 hypersensitivity reactions with IgE and mast cells. But there's also some other intestinal reactions and whatever. But the question is like, how do we not normally respond to the food we eat as foreign? But if we take in an organism, they'll be, reacted against. So one argument would be that it's not dangerous, right? So if you eat something, it's not going to cause damage to your intestine, and that might be what's associated with salmonella is invasive. That's what you would react against. Food, not so much. So maybe that's one explanation. And exactly the why, we don't know. But the how is that somehow there's tolerance to food antigens, generally, unless you develop an allergy, right? And so how does that work? So there must be some kind of antigen-presenting cell that's presenting these antigens in the food that we eat and generating some tolerogenic response. Well, prior studies have shown that this involves T regulatory cells. So there has to be generation of T regulatory cells in the intestine to food antigens. And if you don't generate that, you get an immune response to the antigens, loss of tolerance, and you react to your food, and that's bad. So this paper really sets out to address who are those antigen-presenting cells. Now you can imagine that's kind of hard to do in vivo. Lots of people have tried. They've implicated several different types of antigen-presenting cells in presenting dietary or food antigens. And they've used things like knockouts or reconstitutions or whatever. But all of those are sort of have confounding factors. Like if you delete one subset of cells that you think is involved, another subset could compensate. So there's really no way to say, who were the antigen presenting cells in vivo that were presenting the antigen to drive these tolerogenic T cells.
Right. And knowing who the antigen presenting cells are is particularly important because that may help us understand either how they're driving the regulatory T cells or what this response is that they're driving.
Right. And if we knew that, we could potentially make vaccines or something to drive those production of those cells to help people who have food allergies, for example, right? So there's a couple of different things that we need to know about here. There's a lot of different antigen presenting cells, a lot of different subsets of dendritic cells, for example. There are what we call CDC1s, and these tend to express CD103 and CD11B for the aficionados. And these are a subset that we're going to hear is involved in generating tolerogenic T cells. The CDC2s have CD103, but also have CD11B. And those, at least in this paper that we'll get to, are good at driving Th2 responses. And then there's also a whole bunch of other cells, some of which I had not heard too much about. There are extrathymic air expressing T cells. So ETACs that express air. So air is normally expressed in the thymus to present host antigens to tolerize T cells, but apparently can be expressed in some cells extrathymically. Then that there suggests that they are also involved in tolerizing T cells or making Tregs. These tend to express a gene called ROR gamma T, and they call these Janus cells and Thetis cells, which were a little bit new to me, but interesting. But regardless, so a lot of what has been done before, like I said, is if you think this subset's involved, you knock them out and you test and whatever. But we know that when you knock out one subset, you can compensate. And so there's just some questions around who is actually the antigen presenter who's generating these Tregs to tolerize us against our food antigens.
It's sort of like you don't want to know who could be, you want to know who actually is.
Yeah. So but how do you do this? Like this is what you really want to know is, okay, in the gut, who is taking up that antigen and presenting it to a T cell and making a Treg. But there's like no way to do this in vivo, right? Except that there is this cool technology that was developed in the Gabriel Victore's lab that's called labeling immune partnerships by sort tagging intercellular contacts, or Lipstic. And I wore lipstick today just for that.
Yeah.
So basically you kiss the antigen presenting cell and the T cell. And when they kiss, they leave a mark. So literally just like kissing your finger and leaving a mark of lipstick, it leaves a mark. And so you can figure out who was presenting the antigen. And there's a couple of things that we need to know to be able to understand how this works. So it's proximity labeling. So what happens is they have an antigen presenting cell that expresses something called G5, and they fuse it to CD40, which is a protein expressed on antigen presenting cells, interacts with CD40 ligand on T cells, and helps activate the antigen presenting cell. Okay. G5 is a receptor for a protein called sortase. So they put the sortase on the T cells, and they fuse that to CD40 ligand. So if CD40 and CD40 ligand come together, sortase binds to the G5, and then you get this kiss. And in the presence of a reagent called LPETG, we'll just call it the reagent, and that's conjugated to biotin. It basically, the sortase goes and it plops a covalently attached biotin onto the antigen presenting cell.
Specifically onto the G5, right?
Yep. So now you have the antigen presenting cell that interacted with the T cell is now labeled with biotin. So you can tell who was presenting. So, but how do you get an antigen specific effect of this, right? So the way they do this is they take OT2 T cells. So OT2 T cells are CD4 T cells that are specific for synfecal peptide, which is a peptide of ovalbumin or OVA. Immunologists love OVA. But this is really useful because now you can take T cells that you know are specific for this OVA peptide, and they can have a sortase, and then you can have APCs that have the G5, and then they're only going to have the APC labeled if it's presenting the right peptide to the right T cell. So now you can inject your LPETG biotin, and only the APCs that are presenting OVA to the OVA-specific T cells get labeled. So now you have a way in vivo to mark the antigen-presenting T cells. And so you can feed animals OVA, and the APCs will take up the OVA, and then they'll present it to the T cells. And now you can know who was actually presenting the antigen to the T cells in vivo when they're generating the tolerizing response.
And it's also pretty nice because, OVA is not something that you could also have any sort of self responses to,
right.
Or you're not going to have an infection with chicken egg. And so it's pretty specific as a food antigen in this case.
Right. And we do eat eggs. I had some this morning, so we do eat ovalbumin. So it actually has a relevance to human biology. So one other piece that you need to know is that you can feed ovalbumin and cause a tolerance to OVA. And so you can set up a model system where you challenge mice with ovalbumin plus cholera toxin, and this will sensitize them to OVA allergy anaphylaxis, right? So this is like if you eat OVA and you have an anaphylactic reaction, blood pressure drops really quickly, and the animals will die. And in fact, in this model, 100% of the animals are dead in 20 to 30 minutes. So it's super fast anaphylaxis. And if you just give OVA and cholera toxin, and then you come back and you give OVA, the animals die. If you feed them OVA orally a couple of times, and then you try and sensitize them and induce the anaphylaxis, they're protected. So something about eating the ovalbumin generates a tolerogenic something, right, that protects the mice when you try and induce the anaphylaxis model.
Yeah, it's sort of like, was the first time you encountered ovalbumin the normal way without cholera toxin, or was it the dangerous-y way with cholera toxin?
Right. And that comes back because dangerous might take different forms. So we'll come back to that. But basically, if you do this OVA cholera toxin and you sensitize the mice and then you challenge them, they have high levels of IgE, and they have, they have an anaphylactic response. If you pretreat them with OVA, they have similar levels of IgE, but they have FOXP3 positive T regulatory cells in the gut. So suggesting that these generation of these T regulatory cells happens when you feed just ova, and that can then protect you from an anaphylactic response. So we knew that there was this model in which you could generate anaphylaxis. We knew that dietary ova feeding protects from that anaphylaxis. And this group had this model to be able to see who's presenting antigens. So now they could connect these two, and that's really the big advance of this paper. And then they add in another layer that we'll get to in a minute. But basically, they set out to show whether they could use Lipstic to identify which APCs are presenting the antigen to the Tregs and generating Tregs, which subset. So they actually find multiple subsets that can present the antigen, but which ones actually generate the Tregs. And then they add on a layer of this, which I thought was interesting, about helminth infections. And we'll come back to that in a couple minutes. But the bottom line is that they could label these APCs in vivo, and they found that there were specific subsets that were labeled, and there were a subset of those subsets that actually generated the T regulatory cells, and that helminth infection affected this. So let's dive into what they actually did in this paper. So the first thing is they have to ask, does this Lipstic thing actually work to label APCs that are presenting dietary antigens? So they took these G5CD40 genetically engineered mice that express the G5 on their APCs, and they do adoptive transfer of OT2 T cells from the sortase labeled CD40 ligand mice. So now they have these mice that have their APCs that are ready to be labeled and their T cells that are ready to label them. And a day later, they either feed them ovalbumin or PBS by gavage. And then a couple of days later after that, they inject them with this biotin labeling reagent, LPETG. And then they collect lymph nodes 2 hours later, and they say, are there any antigen-presenting cells that are labeled with biotin? And the answer is yes. So they could detect these biotin-labeled APCs. They were CD11b positive, which is interesting. And they could only see them in ova-fed mice, but not in PBS-fed mice. So something about the oral tolerance to the food antigen is generating these cells that can present the antigen, right? And I'm sorry, that they were only in the ova-fed mice because they're the only ones that had the antigen. Then they also demonstrated that this was really specific because they could inject anti-CD40 antibody. So that's going to block the CD40 ligand interaction, and that blocked the labeling. So this all made sense. It seems like, yes, they can set up the system, they can feed ovalbumin, and they can tag the APCs that are presenting the food antigen. They also asked, does this require MHC? Because one would think it should require MHC. You have to take up the antigen, you have to chew it up, you have to present it in the MHC, and that's how the T cell sees it. So to do that, they used what's called a mixed bone marrow chimera, in which there was 75% of MHC positive and 25% of MHC negative. They all were labeled with the G5. So they were genetically engineered to have the receptor. And they did the same exact protocol where they fed the ova, they labeled, and then they looked by flow, and only the cells that had MHC were labeled. So again, this is telling them that the system works, it requires antigen presentation, it requires the antigen, and that they could see specifically the biotin labeled cells after feeding the antigen.
I wonder why they did 75-25.
I was wondering about that too. I don't know. Maybe because I don't know why they didn't do 50-50. I don't know. Actually, I wondered that myself. I don't have an answer for that.
Maybe it's way harder to get MHC deficient bone marrow.
It could be. And they just did the best they could.
Yeah.
Yeah, I don't know.
Oh.
Okay, so they have these biotin labeled cells that are APCs, and they asked, could they induce T cell proliferation? And it turns out that yes, they could. So they isolated the APCs and they incubated them with CFSE labeled OT2 cells and waited 96 hours. And they looked, and the total dendritic cells, so all of the APCs, and the biotin negative DCs induced very low levels of T cell proliferation, so they could do it. The biotin positive APCs induced a really high level of proliferation, and they also induced FOXP3 expression. So suggesting that if you isolate out the biotin labeled APCs, so those were the ones that we know are presenting the antigen in vivo, and you take them out, you put them in culture with naive T cells, they can actually generate FOXP3 positive T regulatory cells. So that's kind of cool. But they were like, okay, so we know they're biotin labeled, but what are they? What kinds of dendritic cell are they? There's a lot of different kinds of dendritic cells. So they do a little bit of playing around with ratios of CDC1s and CDC2s. So those are the two dendritic cell subsets they're particularly interested in. And they looked at the ratios of them along the intestine, and turns out there's a lot of CDC1s in various different parts of the intestine, including the duodenum, where they think that that's a major location of antigen uptake for generating T regulatory cells to food antigens from prior studies. So they use the same setup to label the cells, and they did flow cytometry for CD11C and MHC2 to identify resident versus migratory dendritic cells. So resident cells have CD11C in high levels and MHC low, whereas migratory cells have CDC high, MHC high. So they're able to separate those out. And I guess it had to do with, are they resident APCs in the lymph node that are taking up antigen that's draining into the lymph node and presenting it there? Or are they actually dendritic cells that probably took up antigen in the gut and then migrated to the draining lymph node because they're isolating the draining lymph node? And what they show is it's actually the migrating cells that lit up with biotin. So it's the migrating cells that came from the intestine to the lymph node that are involved in presenting the antigen and driving the T regulatory cells.
And that makes sense because you probably aren't going to necessarily see the free antigen trafficking. It's much more likely to think about the cell trafficking.
Yeah, I guess, I don't know, because both happen, right? Because even if you inject something like a vaccine in your arm, some of the antigen is going to drain directly to the lymph node and some is going to be picked up and brought to the lymph node. And I guess there's an idea of maybe the way the antigen is perceived, whether it's brought in or drains in, and who's handling it might change the outcome of the response.
Yeah, I was thinking more about the intestine and whether or not the free antigen would be leaving the intestine.
Yeah, I don't know. I mean, there's leaky gut after every time you eat, right? So your gut gets a little bit permeable every time you eat, postprandial leakage. So antigens theoretically could go in too directly. But it does seem more likely that they would be taken in. Okay. So basically what these studies showed is that although both migratory CDC1s and CDC2s, they showed this, could take up and present antigens, it seems to be the CDC1s that are generating the regulatory T cells. They also did a thing where instead of just gavaging, they fed it. This was probably something that a reviewer requested, that can you just put the ova in the food and see the same thing? And yes, they could. So that was kind of cool. So they wanted next to figure out what are the characteristics of these dendritic cells that are presenting these antigens and generating these T regulatory cells. So they do the standard thing, which is single cell RNA-seq. So to do that, they collected the biotin labeled and the non-labeled after the Lipstic model, and they do single cell sequencing. And I'm not going to go through all of the different analyses they did, but basically they plot all of the cells from all of the different conditions and then start to look at, are there clusters and what are these clusters, and are there preferential, distribution of these clusters in certain treatment conditions or in certain populations. And what they find is that there were were 4 clear clusters, 2 migratory and 2 resident APCs. And they used CCR7 and MHC2 to define subsets this time. And when they do that, it's within the migratory DSAs, they observed CDC1s and CDC2s. So they had both CDC1s and CDC2s in the migratory cluster. They knew the migratory cluster was the ones that were presenting the antigen and driving the T regulatory cells. So both of these, CDC1 and CDC2, upregulated CD80, CD86, and CD40. So they're capable of activating naive T cells. But interestingly, the CDC1s had higher expression of genes that are associated with generating Treg cells. So that's kind of cool. So it's probably the CDC1s that are doing this. So what are some of these genes? ALDH1, A2, TGF beta, integrin beta 8, and COA7. I don't know what that one does, and SOD1. The CDC2s, on the other hand, express genes that are associated with T cell activation. And they confirmed all of this by flow. So now they suggest, based on these transcriptional profiles, that it's actually the CDC1s that are in fact generating the T regulatory cells, even though CDC1 and CDC2 within the migratory cluster could both present the antigens. Okay. So next, they say, okay, so in theory, it looks like these CDC1s can be the ones that are generating the T regulatory cells, but can we test this? So they isolate the biotin positive and biotin negative cells from their model and then culture them in vitro with naive OT2 cells with or without stimulatory peptide. And only the CDC1s could induce FOXP3 Tregs. So it's true that in vitro also, in an isolated reductionist approach, only the CDC1s generated FOXP3 positive T regulatory cells. But interestingly, and this kind of fits with, the idea that we're going to come to where there's multiple different kinds of APCs, is that the CDC2s did induce what's called a non-canonical hypo-responsive T cell subset that I had admittedly not heard of before. That's called FR4 positive Th-lin negative cells. Had you heard of those?
No.
Yeah, I didn't know about them, but I looked it up and there are a couple of prior papers that suggest that those cells, and I believe they express ROR gamma T, are actually involved in this regulatory T cell generation in response to food antigens. So the idea was that these were previously described ones, and they might also be important, but this group argues the CDC1s are more important because of their tagging system and who actually could generate more of these T regulatory cells. So CDC1s could generate some of these FR4s, but not too many. The CDC2s could generate a few more. None of these generated TH1s, TFHs, TH17s, or the other kinds of cells. So the CDC1s could generate the traditional Tregs, and both could generate some of these FR4 cells. So now it's a little complicated because there's more than one type of APC that could generate some cells that have some sort of T regulatory function.
Yeah. So you mentioned before the experiment where they were putting the ova in with the food.
Yep.
And I really like that experiment, thinking about this, because I could imagine you might be able to then ask a question in the future of, okay, so are different types of diets going to push different types of
great question
of these antigen presenting cells? So if you gave the mouse now a high fat diet or a fiber free diet or whatever difference from normal chow you wanted to do, could you actually see sort of a push on those antigen presenting cells away from these CDC ones that are making Tregs to other kinds of antigen presenting cells, or to now have those cells inducing other types of T cell responses? And so, they also think a little bit about this pushing response being different. But I think that that's a cool experiment that could be done in the future with this idea of can you then now alter this Treg formation?
Yeah, I love those experiments. I think that's a great idea. And if they're listening, they should do this. Because you're right. High-fat diet affects macrophages, chronic inflammation. It changes the types of cells that you're going to make. Yeah, it's all of that. It's really interesting, especially like if you have some of those carbohydrates or short chain fatty acids that we also know modify APCs and their function. Yeah, it would just be really interesting to look at different diets, because that would be something that's modifiable for humans, right? So we could look at, could you add or subtract things from your diet that might influence the generation of T regulatory cells?
Exactly.
Yeah, cool. So, the fact that these guys are implicated in generating Tregs and they can do it in vitro, they wanted to ask in vivo. And I'm not going to go into these too much, but basically what they start to do is what they called intersectional genetic approaches, which I thought was funny the way they said that, because it's not straightforward to just knock out one of these populations. That's how I started on this, because that's the caveats to most of the prior studies, is that they've had to like knock out one subset and then another subset might compensate. And we'll see that this actually does happen, but they kind of take advantage of that. So the first thing you want to do is try get rid of the CDC1s, right? Because if you can get rid of the CDC1s, then you could to potentially say, oh, that they're the ones that are important and close the loop. But there's a caveat to this. So they use a model system where there's a floxed MHC2. So you can knock out with Cree the MHC2, which is fine. And they use CLEC9A, which is preferentially expressed in CDC1s. The caveat to this experiment is that it's also expressed in some CDC2s. So when they used this model, they did see a reduction in the Tregs by about 50%. So CDC1s and probably a little bit of CDC2s are important. Then they try to use a reverse approach where they're trying to knock out the CDC2s. And that's kind of complicated because they use a ZEB2 enhancer triple mutant called Delta 123. And apparently that's more specific for CDC2s, but not CDC1s. And it's really complex, and I didn't quite understand why they had to do it this way, but they had to do a bone marrow transplant with these Delta 123 cells into wild-type mice to create CDC2 deficient mice. When they did that, though, basically the bottom line is that there was similar generation of Tregs, so it doesn't seem to be these cells. But there was reduced of these FR4 cells in both of those two models. So they argue that those are much less important for generation of food tolerance, these FR4 cells, even though other groups have already suggested they're important. If they knock out mostly CDC1s, they still see FR4s. If they knock out mostly CDC2s, they see FR4 cells. But there's a different sensitivity to the generation of Tregs in these two models, where if you knock out CDC1s, you really reduce Treg induction. You knock out CDC2s, basically the same Treg induction. So it seems CDC1s are really the important population. So up to this point, they now say that it's the CDC1s and CDC2s that pick up antigen. They migrate to the draining lymph nodes. It's the migratory population. They can present these dietary antigens, but it's the CDC1s that seem to be important for inducing the T regulatory cells. They go off on a little bit of a tangent by saying, okay, so when we knocked out the CDC1s, there was still about 50% left. So there must be other cells involved in generation of regulatory T cells. So the only reason why I'm raising this experiment is because they use something they call universal lipstick. So they come up with these cool names. So this puts the, so the sortase is expressed on all T cells. And basically it's going to label any APC. So let me see if I get this right. The sortase expressed in all T cells under the control of the CD4 promoter. And Cre-negative cells, so all of the other cells besides the CD4 cells, express G5. So basically all the cells express G5 except the Cre-labeled cell, the Cre expressors, which are the CD4 T cells, and they express a sortase. So now you've got the system where you can label anybody who's interacting with anyone.
Anybody who's interacting with a CD4.
Anybody's interacting with CD4. That's a better way of saying it. Yeah. So they found that in 14-day-old mice, there was a bunch of different APC populations that were labeled, including some of these ROR gamma T positive cells. And long story short, these ROR gamma T positive cells go down as mice age. They are labeled less. So they think they're really important during early development, which is also interesting if we're thinking about when kids develop allergies and when they're exposed and which kinds of cells are presenting antigens. So it may be that these ROR gamma T positive cells are really important early, but they go away, and almost 95% of dietary antigen presenting T cells in adults are these CDCs, these CDC1s and CDC2s.
I mean that's particularly important because they of that point we made earlier about this involving the first time you see the antigen.
Yep.
Which is likely going to be during that phase when you're younger.
Yeah. Unless we keep kids from eating any peanuts. And that's a whole nother thing. Like, should you be exposed early or not? But
I have a naive question.
Yes.
So all of this is done with ova.
Yes.
Right.
Mm-hmm. Yeah.
So do we assume that other foods are going to use the same pathway?
We do, based on this, that other foods would use the same pathway, although they did not show different antigens. There's one experiment a little later that they get a little bit at presenting multiple different antigens, but to answer a different question. So really they're relying on this ova. Yeah. And I would argue that we eat eggs and ova is in eggs. So and people do develop egg allergies. So in this case, it's actually a relevant model where normally oval albuminitis in mice is not.
Yeah. I think it's also important that if we're wanting to study sort of this CD4 T cell response, we probably have to look at a protein antigen. And so OVA is a nice model there. But and in fact, those exact questions, Vincent, are why I was thinking about that thing where you added OVA to other types of diets to look at diets or meals or whatever that have predominantly other types of nutrients.
Yeah.
I mean, I don't doubt that OVA is relevant. I just wonder if it's all OVA, if it's all correct for all other foods, or if there are going to be differences. I can imagine there would be differences, right? That's the immune system.
Yeah. Yeah. Okay, so they shift gears at this point, and I actually wondered why these two, that up to this point would have been a nice story for me, and they add a whole nother dimension with helminths, worm infections, and put it all into one paper, I guess, to get it into a higher journal. But I almost thought they were two different questions that they were asking. But anyway. So
I look at this as now they're trying to do that thing where they try to push something other than Tregs.
True. That's true. So what do we know about worm infections? They happen in the gut, and they tend to drive Th2 type of responses, not so much Treg. So this is an opportunity to say, hey, I guess for me, it's opportunity to say, hey, if you have an infection on board, how does that influence the generation of T regulatory cells? Now, I'll say that this group and other groups have already done several studies looking at this with different infection models. So prior studies showed that both bacterial and viral infections interfere with Treg generation to dietary antigens. So generating Th1 or highly inflammatory responses while you're trying to generate oral tolerance disrupts the generation of oral tolerance. This group also showed that helminth infection with Strongyloides venezuelensis, I don't know if I said that correctly, which targets the duodenum, induces migratory CDCs that look like Th2 drivers, and they drive Th2 cells at the expense of Tregs. So there's more Th2s, fewer Tregs. And that when they test oral tolerance in those mice, they have reduced oral tolerance. So all of that they knew already. And they argue that, well, let's look at another helminth and see if it does the same thing. So that's why I thought this is a little bit weird for this paper. But anyway, they chose a different worm that's called Heligmosmomoides polygyrus, or H. polygyrus. I'm not going to say that whole word again. And this infection infects the same part of the gut. But I think why they did this and what's interesting is H. polygyrus establishes more symbiotic, like chronic infection rather than an acute infection that's cleared. So strongyloides tends to infect strongly, peak at day 8, and it's totally cleared by day 18 or day 14. So you can look at during strongyloides infection whether or not, you're at the peak of infection or you're after the infection is cleared, and is there an imprint? How does that affect? Whereas with polygyrus, you're kind of looking at it goes up, kind of stays up long term. And you can ask, is there a difference in the ability of these two organisms and their infections to interfere with generation of oral tolerance? So the first experimental setup that they use is that they infect mice with strongyloides, H. polygyrus, or no infection. Then they wait 5 days. So they're kind of in that peak of strongyloides, but basically a chronic infection with polygyrus. Then they adoptively transfer their OT2 cells. And then they give 2 doses of oral ova, which in the uninfected mice should generate tolerance. And then they look at the induction of Tregs. And what they see is that with strongyloides infection, basically Tregs go down to almost nothing. But there's very little change with H. polygyrus. So that's kind of interesting. Then they ask, okay, well, there's still Tregs in the H. polygyrus, and there's almost no Tregs in strongyloides infection. Is there a difference in their sensitivity to ova and anaphylaxis? So that they do that same model, and then they do their ova cholera toxin sensitization, and then they do the IP ova and see whether the mice die. And in uninfected mice that get cholera toxin and ova and then challenged with ova, all the mice die. In the mice that get oral ova and then they get cholera toxin and ova, followed by ova challenge, all the mice survive. So they developed oral tolerance by getting fed ova. If the mice were infected with strongyloides, and then they get the oral ova, the peak of the infection, then they get the ova cholera toxin to sensitize, and then challenge, they were no longer protected. So the conclusion is that strongyloides infection interferes with the ova tolerance. So they lose their ova tolerance. And when they do it with the HP infection, the mice were still protected. So their conclusion is that every infection is not the same. And, whether you're chronic infection or acute infection and the level of Tregs you induce is different depending on your infection. So they went on to show that if they allowed full clearance of strongyloides, so if you let strongyloides infect and clear, and then you do the oral tolerance and challenge, the mice were protected. So it's something about the concurrent infection that disrupted the ability to induce Tregs. So the idea here is that at least for some infections, including some bacterial infections and viral infections that have published previously in strongyloides, if you are infected, something about that infection disrupts your ability to generate tolerance to dietary antigens. And I think that's important because maybe some kids that generate allergies to certain dietary antigens, maybe they had an ongoing infection when the first time that they were exposed.
Yeah,
we don't know.
I think that that's a model that I've seen in some other systems. Particularly, I think it's can't remember if it's murine norovirus or if it's rotavirus in the celiac model.
I think it's rotavirus. I think I saw that. Yeah.
I know that there's that model, and I think that this idea of if it so happens that you are unlucky enough that at the point where you are trying to make antigen-presenting cells that are supposed to make Tregs, if something else is going on in your immune response so that you can't make Tregs correctly, then you're an unlucky allergy sufferer.
Exactly. Yep. I agree. So they did a few other experiments. One other cool thing that I thought was worth talking about. They wanted to look at the Treg induction following infection and with or without the ova and everything. So they used a cool fate mapping approach. So if you like these kinds of things, they called it an I-cell tomato model. And what happened?
I sell tomatoes and lipstick.
Yeah, exactly. So in this model, naive T cells express CD62 ligand, which is the cell gene, SELL, and they are permanently labeled with tomato because they are, if you give them tamoxifen. So I is inducible cell, CD62 ligand. So those cells that are expressing the cell gene at the time that you give them tamoxifen will permanently be tomato positive. So permanently red. And then you can give them a bolus of this and you can tag cells at a moment in time. Any cells that arise after that are negative. And any cells that got tagged, they were naive at the time. And now you can do whatever you're going to do to the mouse. And if the T cells get activated, they downregulate CD62 ligand, but they stay tomato positive. So now you can distinguish the cells that were naive and are now activated because they're tomato positive, CD62 negative. And you can see the cells that were naive before and are still naive because they would be positive and CD62L positive. And then you could see the cells that either came before or after. I mean, if they came after, so they would be tomato negative. So you can distinguish all of these populations, and then you can do infection. And what they see is that 40% of the tomato positive cells are CD62 negative, CD62L negative, and basically they're also GATA3 positive. So they're likely Th2 cells. So there's a very strong induction of Th2 cells with strongyloides infection. But the cool thing is that when they looked for FOXP3, they found that Tregs generated during the infection, so they would be tomato positive, CD62L negative, FOXP3 positive, were just much fewer than those found in uninfected mice. So that's really they're nailing it home that the infection really impairs new generation of T regulatory cells when there's an ongoing infection. So I thought that was a pretty cool experiment. And they
That is a super cool experiment.
Yeah. They go on. This paper goes on. There's just so much there. And a lot of these are really complicated experiments, but a few other things they found was that strongyloides infection, there are far fewer CDC1 cells. And those that were generated were just not able to induce T regulatory cells. So even though they had some CDC1s after strongyloides infection, they just were impaired in their ability to generate Tregs. Then they also did this really complicated experiment, so I'm not going to go into the details, but it comes back to what you were talking about with other dietary antigens. So they co-culture their biotin-positive APCs. So that means those were the ones that were presenting ova in their model. They tagged them in vivo, and they cultured them with a different antigen in vitro. So they would take up that second antigen, chop it up, put it in MHC, and then they expose them to the T cells that are specific for that second antigen. So we knew those ova, those APCs were expressing ova to T cells in vivo. We took them out, we feed them a new antigen and give them the T cells, and we say, can they make Tregs? And they do. So that means that these biotin positive cells can generate T regulatory cells. And they could do that still if they came from polygyrus infected cells, but they could not if they came from strongyloides infected cells. So the conclusion from this is that's really an intrinsic activity of the APCs that are acquiring, presenting the dietary antigen in vivo. So that I thought was really
sort
cool.
of intrinsically, did they see danger?
Yeah. We still don't know what happens during that time, although one of the experiments, again, I'm not going to go into too much detail because this is already long, but they found that the imprinting happens in the intestine. So they did an experiment where they looked at the cells that were moving into the lymph node versus the ones that were in the lymph node. It's really some sort of imprinting activity that happens when the cells are in the gut and they take up the antigen. And that way, when they move to the lymph node, they're presenting that antigen and generating T regulatory cells. So it kind of comes back to that idea of cells that are imprinted in the gut come back to the gut and have a unique type of functional outcome. Lastly, they did an experiment where they tried to say that it's really this ratio of CDC1s and CDC2s that's important. That if you lose your CDC1s and there's lots of excess CDC2s, we lose the ability to generate Tregs. So I don't think this was the greatest experiment, but I think it was the best one they could do with what they had. So they used the Delta 1, 2, 3 mice, because remember, they knock out the CDC2s. And if you infect with strongyloides, there was a massive number of CDC2s. And they're just going to basically knock that down and make it about 1-to-1 ratio. And they showed that basically they got 1-to-1 ratio. So that's good. So then they had a system to say, okay, what if there's equal numbers of CDC1s and CDC2s during the infection? Could you restore some of the T regulatory cells? And the answer is yes, partly. So they restored more Tregs, fewer Th2s. And when they tested an anaphylaxis model, they were now protected. So they argue that there's the strongyloides infection particularly biased really strongly towards a huge number of Th2 cells, including generation of CDC2s that mediate the generation of those Th2 cells, and that this impairs maybe the migration or generation or proliferation of CDC1s. Therefore, you have fewer CDC1s, less T regulatory cells, and in this case, of ova, less Treg generation is the loss of tolerance to the dietary antigens. So that was the whole paper. It was really complicated, but I think there's a lot of interesting pieces around this. One of the things I was curious about is what you guys thought about the hygiene hypothesis, because this idea is that the more you're exposed to parasitic infections during early childhood, the less likely you are to have allergies or asthma. And this paper is actually suggesting the complete opposite, where if you have infections, actually more likely to not have tolerance to food antigens, develop allergies. They also used an asthma model that I didn't go into, but they showed that if you tolerized, you could prevent the asthma induced by the ova too. No, what do you guys think about that?
So I think that my overall view on the hygiene hypothesis has been evolving.
Yeah.
And so, so I think that maybe the original idea of just infection protects you, not being infected doesn't protect you is a bit of an oversimplification.
Yeah.
I know that we talked in a previous episode to Kiki Fairfax. And I know that she and others have thought about things more as like the old friends hypothesis of our immune system evolved to work one way, and now we've sort of pushed it in a slightly different direction. And so that's been where my understanding has started to go. And so here, I think that maybe the idea is that it's about when you're first encountering that food and how we evolved to first encounter food versus different types of infections. The timing, in theory, you are probably not chronically infected with something when you're very young. And so that might get to the sort of chronic versus short-term infection with the different helminths. But I just think it's a lot more complicated than all of that, I guess, is my short answer.
Yeah, I would agree.
I don't know that most people get, at least in the U.S., get parasite infections at a young age, right?
No.
Right, yeah. And so that's been the idea, is that we see more sort of allergy and certain types of autoimmune diseases in Western countries. There's a bunch of different epidemiological things that are linked to protection from allergy. And so the hypothesis was, well, if you get a parasite, that protects you from allergy. And one of those counter hypotheses hypotheses is if you get a parasite, it generally immunosuppresses you, and your immune response is not going to ever be high enough for you to have an allergy. And so I think that there's the original epi suggested that the infection was protective and that was it. And now we've kind of made our understanding of immunology a little more nuanced, shockingly for immunologists.
Yeah. And I think that this would say that an infection at the same time that you're exposed to an antigen for the first time will influence how that antigen is perceived by your body.
Yes.
Which is interesting. But I also wonder if it's types of antigens. You asked this, Vincent, is it just ova? It's probably not just ova, but are there characteristics about certain antigens that make them more likely for this to happen? So if you had like P protein or something that most people are never allergic to, and we tested that in this model system, would it also fail? Or is there something about some of these antigens that are common allergens, like eggs or peanuts or shellfish, right? That somehow they are handled differently. I don't know if it's their solubility or the way they're presented, or I don't know. Yeah.
I mean, the other sort of similar question related to all of this, and this is in no way a critique of this paper because I don't entirely know how you would do this experiment. But this is really thinking about oral tolerance. And so what would be really cool is if there could be a comparison to if you were getting ova through the skin, do you also tend to have CDCs presenting? And do they also make the same Treg? And so is it a difference in the type of antigen presenting cell? Because here in this paper, they've really identified who is the antigen presenting cell.
Right.
So is it a different antigen presenting cell in this anatomic location compared to others? Or is it a difference in what happens in the draining lymph node after the antigen presenting cell is presenting the antigen?
Yeah. So I'm assuming there's probably a way to do this. You can probably label the dendritic cells with G5 that are in the skin versus the gut or something like that. You can put it under some control so that you can label specifically the ones in the skin. Or we can even just paint on antigens and collect their draining lymph node and ask what's going on. But it's interesting. And I think it could be used, I think they've already used it somewhat for vaccine responses. So which are the APCs expressing vaccine antigens? That's important. Yeah.
Well, my food is digested. I'm good.
And I have to tell you, this discussion has made me hungry.
So some people eat before and after. I know it was a long discussion, but I thought there was a lot of really cool experiments they did in this paper with cool tools that were worth going over.
The experimental tools are amazing.
Yeah. It's really cool.
I love seeing how, people are kind of being able to innovate and ask different questions and think a little outside the box with these really cool techniques.
Yeah.
So, Brianne, you want to have some eggs?
I can have eggs. That's fine.
Okay. It's Immune 89. You can find the show notes at microbe.tv/immune. And if you'd like to send in questions or comments, immune@microbe.tv. That's an email address. I know a lot of people don't use email anymore, but we still do. And if you enjoy these programs, if you learn from them, please support us, microbe.tv/contribute. Cindy Leifer's at Cornell University. On Threads, Cynthia Leifer, one word, and on Blue Sky, Cindy Leifer. Thank you, Cindy.
Thanks.
Brianne Barker's at Drew University. I don't have your Blue Sky here.
Oh, BioProf Barker.
Oh, that's BioProf Barker on Blue Sky. Got it. Thank you, Brianne.
Thank you. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.